Uh, Formal topics. The first is the buffering action in blood. This is a famous theory, and this is actually from where buffer got a big fame. Buffer solution, one of the best performing buffer solution is blood. It's a mixture actually. And as our concept, we always remember that buffering is a limited performance, and in a limited quantity, if something goes in blood and changes the pH, blood is there to restore its pH. Medicines, food, and other things are there. Everything has a pH, and it's a very interesting thing to understand that taste, the taste of many things which we like a lot. And it's a very funny example that what happens when you see something very delicious and you like it, you feel the pH change in your mouth. And this is a remarkable working of a buffering system when it first allow this change and then it retains it. Similarly, when you have a taste of eating something sour, some people like salty, and you put it in your mouth and you feel the signals to the brain, and this is again a pH change. So as mentioned earlier, that buffering is actually a restricted fight against pH. But the important point is there, pH will be allowed to change, then it will be restored. That is a miracle. Why? Because if pH doesn't change, then we can't actually differentiate between taste. Like if you are eating a sandwich or something which is in a mixed taste, then obviously we have to feel it. Buffering action in blood. The, the pH of blood is between 7.35 to 7.45. And this changes below 6.8 and 8.0 can result in a death lethal issue. So major buffer system in the body fluid is carbonate buffer, H2CO3. Now, why we scientists believe it's a miraculous design? Because blood is an independent solution and body controls it from itself. So it has to make a buffer itself and it has to maintain the buffer itself. So I'll give the heading, buffering action in blood, and then you have to copy this screen.
Okay. I've copied.
Carbonate buffer. Carbonate buffer works with CO2. And as mentioned earlier, when human body works in a system and the raw materials are needed and extra things have to be removed, body has to work on its own. There is no interference possible from outside until the problem is visible and some injection or something like that. So some CO2, the end product of the cellular metabolism, you know how CO2 is coming, the famous, because you are a bio student, so no need to remind you that CO2 is the product of that metabolism when energy is produced and the breakdown of carbohydrate, etc. So it is carried out lungs for elimination and the rest dissolve in body fluids. So this is where body is having a superior design and where all CO2 is not exhaled. While initially it was thought like that, but some CO2 dissolve in body fluid, forming carbonic acid that dissociates to produce bicarbonate. and hydronium ion. Hydronium ion is also called oxonium ion. So this is how this system works. Now you have to copy this.
Okay. Yes, I've copied.
So this is the buffer in our blood. When excess acid in the body is neutralized by this one, the equilibrium shift left. So suppose something goes in the body which is acidic, like suppose you are eating vinegar a lot, some people have a taste of using that. Different food items are there across the world. And I always tell my students that food industry, like some people think it's just like food, but it is also like cosmetic. It's a huge industry. Many people regularly go to the restaurants and other ways they like some food. But when you start learning the chemistry there of different food ingredients, then you see how chemistry is actually very effective. You shift the pH to create a taste, and you can also introduce some ingredients to generate an artificial taste. So this is a big science, like bakery, from starting from bakery, some cooking, and the preserved foods. Then this is all very important, because without buffering, a food can be in a problem. That is why after a certain time, you say food is rotten, and in bio and chemistry, you say now it's a denatured substance and you can't eat it. So in our blood, oxonium ion in the body is there, which is neutralized by HCO3. What is oxonium ion? Remember, oxonium ion indirectly represent an acid, because acid gives H plus. H plus react with water to form H3O plus. So I repeat, when we say acid, it is normally people think H plus, but actually H plus is too small to stay in a solution. It immediately react with H2O and form H3O plus. H3O plus is actually representing acid. You have to copy this screen.
Okay. I've copied.
When a base enters, again, alkaline. Some alkalis are obviously very dangerous, and you know from chemistry that many people think acid is dangerous. But acid and alkalis are equally dangerous. They are highly corrosive. But there are some alkalis which are not dangerous, and that is why in our beverages, soft drinks, we use them, and they give a sparkling taste.
Yes.
Excess OH minus has to be tackled down by H2CO3. H2CO3, shift the equilibrium to the right to balance absorb the OH minus. We have to copy this.
Okay. I copied.
Throughout our bodies, chemical reactions are rearranging atoms and molecules to maintain the crucial balance our metabolism demands. If the natural acidity in our blood falls, a molecule of carbon dioxide and water react to form a new compound, carbonic acid. This molecule immediately splits into a bicarbonate ion and a free hydrogen ion, what we know as acid. But this reaction rarely occurs without help from another player in this molecular exchange, the enzyme. Throughout our bodies, chemical reactions are rearranging.
The blood system of an adult person contains about 5 litres of blood, a liquid that carries nutrients and oxygen to all living cells in the organism and removes waste products. One of the most important functions of blood is to provide oxygen for cell metabolism and remove the waste product, carbon dioxide. This process is quite complex, and its effectiveness is strongly affected by the pH of the reaction medium. Therefore, the pH of blood plasma is carefully controlled by the organism and maintained at a constant level of 7.4. The pH inside the erythrocytes is a little lower, about 7.25. Effective control of the pH is possible because blood contains a certain amount of carbonic acid, which is a weak diprotic acid, and also a certain amount of its conjugate base, the bicarbonate ion. The delicate balance between the concentrations of these two species allows the pH of blood plasma and erythrocytes to be maintained at a constant level. Solutions containing weak acids and their conjugate bases are capable of maintaining their pH at a certain stable value. Such solutions are called buffer solutions. The blood system of an adult person contains about 5 litres of blood.
So lungs and kidneys, because again, you're a bio student, so there is no need to copy this. And bio obviously explained these things very well, even though it's a rule in Cambridge that you have to be very careful when a biological detail comes in chemistry, which always come in many positions. You have to be careful. You can't use unique bioterminologies in chemistry, and reverse is also not allowed. Just like in physics and chemistry, people should always try to avoid any overlapping. So if you are a bio student, you can feel the language is not pure biological here. Lungs and kidney play important role in regulating blood pH. Now, this word can be commentable, and if you read bio and in this article, they do not use the word regulating, but chemistry use that. The idea is simple that in blood, you have to maintain the buffer solution. Just like in a swimming pool, you are adding water and you are removing water because water is getting polluted. Similarly, you have to add and remove this thing. The lungs regulate pH through retention or elimination of CO2 by changing the rate and volume of ventilation. This is the concept of controlling CO2. Similarly, the last point is about the ammonium ion and then HCO3 if suppose there is a change, permanent change. Now give the heading other important buffers.
Okay. I've given the heading.
The phosphate buffer system HPO4 2 minus H2PO4 minus plays a role in plasma and erythrocytes. What are erythrocytes?
The red blood cells.
Hmm?
Red blood cells.
RBCs.
Yes.
Okay, good. So that is fine. And that is the point here. And we are discussing the equilibria. It's a reversible reaction, H3O plus and H2O. This is another living buffer in our body, which is there for making this system. As I mentioned, there is a number of buffers in our body, starting from the mouth. That is why I gave you the example that you enjoy a taste, the pH changes, but after a while, the pH is back and the saliva is on its neutral position, and you can interestingly enjoy the same taste again. One of my students asked a very interesting question that if it doesn't come back, it means that we can't eat it again. We can eat it again, but we can't enjoy it. That is why the taste reversal and the stomach pH control, they all are being buffered. That is why when we make cosmetics, we make as a synthetic food nowadays, a lot of chips and everything are there. We have to use this idea of chemical treatment to encounter safely with our body buffer system. So this is a phosphate buffer system. You have to copy this screen.
I've copied.
When an acid enters, the equilibrium has to shift backward, the reverse arrow only, to balance the attack. You have to copy this.
Okay. I've copied.
When a base is entered, the equilibrium shift to the right to balance it. You have to copy this.
I've copied.
The ability of a phosphate buffer and the alka seltzer is a medicine to resist pH changes highlights the physiological role of buffers within cells and in consumer products. This is what I was explaining before, and this is a very deep study, and one of the biggest industries like cosmetics, and this is food industry is also one of the biggest industries. There they actually treat multiple things, and obviously pharmaceutical industry also deal with these concepts. Now we move to our next title, analytical chemistry. It is not the start of analytical chemistry actually. Analytical chemistry is now considered to be a separate unit like organic, physical, inorganic, and analytical. So they have made it a separate unit now. Analytical chemistry is dealing with the separation and identifying of substances. So you have to give the new heading in the same chapter, give the heading analytical chemistry.
Okay. I've given the heading.
Separating and identifying substances is called analytical chemistry. You have to copy this.
Yes. Is there inorganic chemistry in A2?
Yes, a little one.
Yeah. Okay, I've copied.
There is inorganic there, but like mainly consisting of family of transition elements. Transition and group 2. Group 2 is a so-called article because it's small, but transition is a king, one of the biggest chapters in our course coming every year. So transition is making up majority of the inorganic. Now we put the last title of this chapter, partition coefficient.
Okay. I've copied.
Syllabus got updated first in 2002, then it got updated in 2007, then it got updated in 2016, and then it got updated last year to 2022. As syllabus is changing after every 3 to 4 years now, so when you plan your past paper, you have to be very known and you have to be careful about the topics which have been removed, which have been added. before 2016, when you look at the past paper, you will find many topics which are very close to bio, like DNA, RNA, biochemistry, enzymes, etc. In 2016, they have removed all of it and they updated the syllabus to a big degree. Partition coefficient was a topic in that sense. There was a section B in those times. Crossing 2016, that topic now is in the regular area. Why these details are important, something is gone or something is not gone? Because every good student prefer doing past paper at least till 2010. So when you go till 2010, you will at least face two major syllabus changes. And obviously, you know that this topic is there, this topic is not there. Partition coefficient, let's make the concept. First, make the concept that what is partition coefficient. But first, I explain what is partition. So suppose that I have a liquid, say X, and I have a liquid, say Y, and these two liquids do not like each other and they are perfectly immiscible. For example, take one as water. Water is polar. Other as hexane. Hexane is non-polar. So they do not mix. They do not mix. Now, if you know that they do not mix and you put them in a single beaker, what will happen? That they will make a layer. They will make layer and they separate like this. The theory is simple that the top one should be the lighter one because it is going up. So Y and X are now in a layer. In chemistry lab, you can perform this and maybe the teacher will show. Some teachers show A2 chemistry labs also because that will really help in paper 5. That you can put the liquids in a single beaker which are immiscible. When they are immiscible, they will form two layers and which the layers are separating from each other. This can be really observed in some oceans also where water form layers and they do not mix, even they are side by side. Now, this is a pair of two liquids which are not immiscible. Now, introduce third member Z. Z, suppose, is able to dissolve in both X and Y. This is like a mediator, like two people, X and Y, are not meeting, not talking to each other. So they use a person who is a mediator who can talk with both of them. So Z is a mediator. And for example, you take ethanol. Ethanol is an organic compound. It is an alcohol, but it has surprising properties like hydrogen bonding. So it is able to dissolve in water. Being organic, it is also soluble in organic substances like hexane. So I have Z. Now I start putting Z crystals or Z particles here. What the Z particles will do? Some of the Z particles will dissolve in, some of the Z particles will dissolve in Y, and some go through the boundary and dissolve in X.
Yes.
Did you get the point?
Yes, I got the point.
Z has dissolved in both of them. Z has dissolved in both of them. And you can notice from my drawing that Z has dissolved more in Y, less in X. This is a possibility. Z is soluble in both, but definitely solubility can not necessary that it is the same. Z dissolve more in Y and less in X, for example. Now, what we will say that Z has penetrated in between X and Y, and it is now soluble in both X and Y, which are mutually immiscible. This concept for Z is called as partition.
Yes.
Then this is called partition. Partition means that a substance dissolves in between two immiscible substances. Partition means a substance dissolve in between two immiscible substances. They do not mix in each other. So Z penetrate and partially mix in Y, partially mix in X. Now, if you find the concentration of Z in Y and you find the concentration of Z in X and you divide them, you divide it to get the ratio. This ratio is called partition coefficient. This ratio is called partition coefficient.
Yes.
So we make the concept. So we make the partition first as our concept. After giving the heading, give a side heading partition.
Okay. I'm done.
So a substance, we start the theory, we start the story like that. A substance dissolves because the total energy of the solute plus solvent system is less when the solute in solution than when it is not. The basic idea behind it, all natural reactions are exothermic. Nature supports exothermic reactions. Nature do not support endothermic reaction. Exothermic reactions have a high probability of being spontaneous. Obviously, Gibbs free energy has to make the test. So substances dissolve like this. This tends to be the case if the interaction between the solute molecule and those of the solvent molecules are similar to those between the solvent molecule themselves. So what it means actually, like for example, how ammonia dissolve in water. Ammonia and water molecules form hydrogen bond. This is called intermolecular interaction. So ammonia happily dissolve in water. I ask you, for example, how an acid dissolve in water. You will say dissociation. How an ionic compounds dissolve in water. You will say reverse of lattice formation and then reverse of lattice formation, reverse of lattice formation and
Sol-
what was the second
Solvation.
step of ionic compound solvation?
Hydration.
Hydration, very good. So how an ionic compound dissolve in water, how an ionic compound, so first reverse of lattice formation, lattice breaks, and then hydration. So these are the important factors. These are the two important factors for solvation of ionic compounds. Why some gases dissolve in water, they just bubble in and diffuse. So this is now in this red area, red lining. This tends to be the case if the interaction between the solute molecules and those of the solvent molecules are similar to those between the solvent molecule themselves. There are four different ways that molecules interact with each other: ion-ion interaction, ion-dipole interaction, van der Waal, and hydrogen bonding. Why we are studying this? Because this is important to understand that some compounds are immiscible, some are partially miscible, some are fully miscible. So story comes from that side. So you have to copy this screen.
Okay. I've copied.
then you have to recall this, you have to copy this.
Okay. I've copied.
So polar solvents are obviously able to dissolve where polarity is there. CI emphasizes here that people should know about the polarity interaction. Non-polar solvents are more likely to dissolve those solute with whose molecules are only attracted to each other by van der Waals forces. I hope you remember that van der Waals forces are always there. There is no question about them. They are the forces which exist between every pair of molecule. When there
Yes.
are no other force of attraction, they are present. And when there are other force of attraction present, they also are present. So this is a concept of polarity interaction. So you have to copy this now.
Okay. I've copied.
Now give the heading partition. This is the idea of partition coefficient, that we have two solutions. Give a side heading partition coefficient.
Okay.
We have water, hexane, and I have bromine. So you have to copy this.
Okay. I've copied.
Now look at the bromine. I have added bromine in this and I have added bromine. Bromine is actually able to dissolve in both water and hexane, but water and hexane are mutually immiscible. So I will find the concentration. You have to copy, after copying this, you have to write under it that water and hexane water and hexane are immiscible. Bromine can dissolve in both water and hexane. You have to copy this.
I've copied.
So we add bromine in water, we add equal amount of bromine in hexane also, and we find that on their concentration in that. You have to copy this.
Okay. I've copied.
So this is called partition coefficient. It has no unit, a ratio of concentration. So KPC is called, partition coefficient is called the ratio of concentration of a solute in two immiscible, mutually immiscible solvents. You have to copy this.
Okay. I got what KPC is, but why do you use KPC? To obtain what?
This is again the idea. You should understand that this topic is coming in this chapter. The chapter is equilibria, and this is again the idea of different industries where we are using solvents. For example, your hands got some organic stain. Suppose you are eating food and some organic stain comes on your hand, and you get up and you wash your hand, but you feel water is not able to remove the stain because the stain is organic. So you take the soap and you rub the soap on your hand. Now, if next time when you rub the soap on your hand, very carefully see the soap and water are actually making a partition on your hand. Some soap dissolve in water and some soap dissolve in that organic stain. Now, after that partition establishment, when you rinse, when you wash your hands, the organic stain go off. This is the idea of partition coefficient. Similarly, if you have a taste of cosmetics, cleanser and many other makeup stabilizers and makeup removers, you also have gone through the ink removers. When we are in our childhood, the ink drops and we have some eradicators which can in liquid form can dissolve. So there are a lot of applications. The idea is there: two immiscible two immiscible liquids, two immiscible substances. So if your hand got some color, if you have a taste of painting, the color comes on our hand. Okay, then you can't remove the color from water. Why? Because color is organic. It's a nitrogen dye, and water is polar, so can't dissolve it. So we need something. We need something in between which can dissolve in water as well as color, and so that color gets in the partition and we can rinse it out.
Okay, I got it.
So this is the boundary, and you can see that this is the theory I had explained you. Is called extraction. That if you have a taste of cooking, not eating, obviously we all are expert in that. But if you have a taste of cooking, then you understand this. And in between cooking, conventional cooking, not talking about the advanced machine cooking, no, technical manual cooking, people keep on tasting. People keep on tasting the substance. And this is again an example of a partition, because our saliva has a partition with that substance, and then we can taste it. Similarly, another good example is solvent extraction. If you have a taste of washing, what should I say? Washing the cutlery and the other stuff after eating.
Yes.
Do you have a taste of that? Have you tried that?
Yeah.
Okay. We have a sponge and we have a liquid, or we have a solid soap. We put some soap on that vim and that foam, and we rub it on water on that cutlery which has some organic stain, and it removes, it makes a partition. This is called solvent extraction. You have to copy this now.
Okay. I've copied.
So this is how we make the partition. Now copy this.
Okay. I've copied.
So this is called partition coefficient. Now, iodine is soluble in two immiscible solvents like water and hexane, and its solubility is very unlikely to be the same in both solvents. So one solvent will be better at dissolving it in the other. So when some iodine crystals are taken with a mixture of hexane and water until no further change takes place, two layers are allowed to separate. So we put some iodine in the mixture of hexane and water and they partition first, and then the ratio of the concentration is called partition. So the ratio of the concentration of iodine in each layer is constant, called partition coefficient. This is an example to understand, another example to understand. So what is partition coefficient? Ratio of the concentration of a solute in two different immiscible solvents when an equilibrium has been established. You have to copy this.
Okay. I've copied.
So partition coefficient has no unit, and useful working out how much solvent we need in order to extract a minimum amount of solute from one solvent into other. Partition coefficient is equal to distribution coefficient. Our syllabus say it like this. You have to copy this.
Okay. I've copied.
So this is the end of this very big chapter, equilibria, and A2 chapters are like that. So the main topic was buffer solution, and we can do some questions in our next class. And also, you can also start whenever you want, you can start this chapter. And like you always do some questions, so you can start doing some questions, and we can also do some questions in class, and then we can, by the time you will be able to ask the teacher what she is planning to start.
Okay.